Good morning, everyone, and welcome to our Research Bites with Professor Tommy Boland. Tommy is Professor of Ruminant Nutrition and Sheep Production in the School of Agriculture and Food Science and is involved in teaching and research activities focused on enhancing the sustainability of pasture-based livestock production systems, enabling students to understand and respond to the myriad of problems facing our global food systems. Tommy is actively involved in communicating science to industry and farmer meetings, national and international scientific conferences, and writes a monthly article highlighting the sheep flock at UCD Lyons Farm in the Irish Farming Independent. He is a council member of the Agricultural Science Association and was recently appointed Associate Dean for Research, Innovation and Impact within the school. So thanks a million, Tommy, for agreeing to give this morning's research bite, and I'll hand over to you now. Thank you.
Very much, Geraldine, and thanks to the Institute of Food and Health for giving me the opportunity to make a short presentation on my research this morning. And I'm going to talk about the what, why, and how of sustainable ruminant production systems. You know, I think it's an area that's been topical for a number of years, but never more so than now as we're discussing carbon budgets and potential changes to how food is produced, not only nationally, but globally. So my presentation this morning is going to focus on ruminants, and ruminants are alloenzymatic foregut fermenters. And basically what this means is that the ruminants have a specially evolved digestive system whereby at the beginning of their digestive tract, the stomach is divided into 4 separate chambers. We often hear it said that ruminants have 4 stomachs. That's not entirely correct. The stomach has 4 chambers, the largest of which is the rumen. And the rumen is home to a very large and a very diverse population of microbes. And the microbes really serve a number of roles, but their most important role is to allow the breakdown of cellulose, which is the most abundant polymer on Earth. And without the enzymatic activity of the microbes in the rumen, the cow and sheep would not be able to break down this source of nutrients. So I suppose that's the really important aspect of the rumen, that they can digest through the activity of the microbes this cellulose, which is something that humans and other monogastric species simply can't do. Now, not all ruminants are the same, and ruminants are present at various stages along the evolutionary cycle. And if we were to consider the most evolved ruminant as being the animal that can consume and digest the largest quantities of this cellulolytic material, then of the ruminants or of the animals we're familiar with, the cattle or the cow would be the most evolved ruminant. So the cow can consume large quantities of feed in 2 or 3 short feeding bouts during the day. And then the animal will spend between 8 and 10 hours of the day regurgitating and rechewing that feed in order for it to be broken down in the digestive system. And that's how our animal production systems have evolved, particularly our pasture-based production systems, where we utilize this ability of the microbes present in the digestive tract of the ruminant to digest grass and other feedstuffs that are referred to as forages. Now, one of the, I suppose, byproducts of this microbial fermentation in the rumen is the production of gases, various gases, but particularly methane, referred to as enteric methane when it's produced in the digestive system. And this is an important greenhouse gas. Globally, about 1/4 of all greenhouse gas emissions are associated with agriculture, forestry, and other land usages. Of this emission associated with agriculture, forestry, and land use, about 60% is from the various livestock systems that are in play globally. If we look at this pie chart, and the obvious major source which stands out is the dark blue section to the bottom left, which represents enteric methane emissions. And this enteric methane, as I've said, it's the methane produced in the digestive system as a result of the microbial breakdown of cellulose and other structural carbohydrates present in the diet. And Ireland then is pretty unique in terms of developed countries globally. I suppose the only other country that would come close to us in terms of our greenhouse gas emissions profile would be New Zealand. And in Ireland, 37% of our total greenhouse gas emissions arise from the agricultural sector. I suppose there's many reasons for this, and I'm going to greatly oversimplify the explanation here by stating perhaps the main reasons are: we do not have a huge presence of heavy industry in Ireland. we actually don't have a huge amount of people in the country, and we have quite a lot of cattle, and particularly cattle in the country relative to our human population. So we get this unusual situation where agriculture accounts for 37% of our total greenhouse gas emissions. And, you know, at a national level and at a global level, this scenario is getting a lot of attention. And, you know, there are various documents which some of you will be familiar with and some of you won't be familiar with, including the Rethink X, I suppose, polemic on, you know, the disruption and the collapse of industrial livestock farming with a particular US focus. We had the Eat Lancet report from a couple of years ago looking at the, you know, the proposed alterations to dietary consumption at a national and a global level. Closer to home, the Climate Change Advisory Council produced a report last week, and then we were seeing headlines like this one on the front page of the Irish Farmers Journal, looking at potential disruptions to the agri-food industry and the ruminant industry in particular from targeted reductions in carbon emissions from the agri-food sector at a, I suppose, the level of job losses, loss to the economy, and reduction in herd sizes. And, you know, this is all, I suppose, resulting from and informing policy decisions at a European level. And the recent farm to fork targets were really quite, I suppose, onerous in terms of some of the changes that are required in our food systems. If we look at this here, we can see a 55% reduction, targeted reduction in greenhouse gas emissions, an increase in organic farming with 25%, a reduction of antimicrobial usage by 50%, a reduction of pesticide usage by 50%, a reduction of fertilizer usage by 50%, and a reduction of the land area dedicated to agriculture of 10%. So these are really fundamental changes being proposed for how we produce food within the European Union. Just to maybe bring it back to a little bit more of a local level, and when we talk about this cellulose and this biomass that's available for, you know, available to ruminants but not available directly for human consumption. If we look at what biomass is required to produce a kilogram of lamb, Elizabeth Earle's work would show that per kilogram of lamb meat produced within the entire sheep farming system, 22 kilos of grass dry matter are required, plus an additional 4 kilos approximately of silage, which is conserved grass dry matter, plus an additional 1 kilogram of concentrate dry matter is required to produce 1 kilogram of lamb carcass or 1 kilogram of lamb meat. Now, essentially, all that biomass resource used to produce that kilogram of lamb meat is unavailable for human consumption, and we can formulate that concentrate feed in such a way that it doesn't contain any feed products which are directly available for human consumption. And I think, you know, that's a very important aspect of ruminant agriculture, is that it serves as an upcycling and a recycling mechanism whereby we can utilize biomass resources not directly available for human consumption to produce products which are suitable and available, and I suppose have very high availability of micronutrients which can be consumed directly by humans. And that's reflected in our, I suppose, our agri-food exports. And each year, Board BIA will produce a report looking at the agri-food exports from the country. And the most recent report was for the 2020 year. And we see that about total agri-food exports, or food exports, account for about 13 billion euros. Approximately 8.5 billion euros of that arises from the dairy and the meat and livestock sector, the vast majority of which is coming from ruminant animals, dairy cows, beef cattle, and to a much lesser extent sheep. So at a national level, in terms of the food we export, products of ruminant origin are really, really important and make a very large contribution to the overall food exports. And why do ruminants make such a large contribution to our agri-food systems here in Ireland? And I think this image paints a really nice picture of what the country looks like on the very rare cloudless days we might get. If we look at the land use scenario in Ireland, the large blue area here represents the area of land in Ireland utilised for grassland. So about 60% of our total land area is utilised for grassland. About 17% would fall under the heading of wetlands, including peatlands, some of which will also have a food production component, mainly through grazing. About 11% of the land area is utilised for cropland, about 11% utilised for forest, which would be rather low by European standards, and then about 2 to 2.5%, which would essentially be settled or urban areas. And if we kind of express the area of agricultural land utilised for grassland, it increases to 90%, you know, so again, a rather unusual scenario, certainly by European standards, where the vast majority of our agricultural area is utilised for grass production. If you look at the topography of our country, you know, maybe it starts to become a little bit evident as to why so much of our land is in grassland. Now, this does also present some challenges, and this map on the right-hand side, it looks at a bovine density map, and you can see that the darker the colorations, essentially the more densely populated livestock are in those regions. And this in itself can present, you know, challenges, particularly from a nutrient loading perspective. One of the ways in which ruminants have evolved to coexist with grazing systems is that they do excrete relatively large quantities of nutrients back out onto the land where they've grazed, particularly nitrogen and to a lesser extent phosphorus. When we concentrate animals in confined areas, this can lead to a concentration of some of these nutrients on grasslands, which can present a challenge from an environmental loss perspective as well. And that's one of the areas where my work is attempting to address and to come up with some solutions. So then just to look at my own research area and what work I've been involved with over the years, I suppose I started off with a very kind of focused component type research approach, looking at the impact of trace element nutrition, particularly mineral nutrition, on the health and of the nutrition of the ewe or the female sheep during late pregnancy, and the subsequent impact on our offspring or on our lamb. I moved on then to look at the role of ruminants in greenhouse gas emissions, and particularly how we might mitigate enteric methane emissions from our ruminants. So it has led to some fairly interesting equipment over the years. Then I suppose expanded that work out into grazing systems with dairy and beef and with sheep. And more recently, and this graphic will be familiar to some of you who witnessed or observed Helen Sheridan's presentation a few months back, to look at this system in its entirety and look at all the the externalities involved in livestock production. You know, so I guess as my research career has developed, I've moved away from the very niche component type research into getting a better understanding of how individual interventions at an animal or a system level can have an impact on the entire food production system that we're operating in. But I suppose just to refer back to some of the work that I've done over the years, and you know, this all links in with sustainability and sustainability of ruminant production systems. And I suppose the first focus was around animal health and animal and lamb mortality in particular. And again, the image on the left will I suppose highlight some of the interesting equipment I've got to use and some of to design in my research work over the years. And we see a picture of a ewe wearing another cover or an apron to prevent the lambs from suckling for a short period of time. So in this work, and again, this is a team that has ran through a lot of my research today, we're working very closely with an industry partner. And essentially this industry partner had a product they wanted to test. And when we tested that product, we discovered it had some deleterious impacts on animal performance. So essentially the product was a mineral supplement which was fed to the ewe during late pregnancy, or the female sheep during late pregnancy. And when we fed that supplement, we discovered that the lamb's ability to absorb antibodies from maternal colostrum was severely reduced. And as the lamb is born completely naive from an immunological perspective, it's essential that the lamb can absorb the colostral antibodies. So over a series of experiments, what we discovered that the iodine, or essentially excess iodine supplementation of the ewe during late pregnancy, reduces lamb serum immunoglobulin concentration. And, you know, this potentially exposes the lamb to an increased disease challenge and potentially to an increased risk of lamb mortality. So once we discovered that iodine was the element responsible, I suppose the problem could either be arising within the ewe in terms of the colostrum she produced, or within the lamb. So we designed what we thought at the time, and actually still do think was quite a nice experiment. We had a number of twin-bearing ewes, so each ewe was pregnant with 2 lambs. One group of ewes were fed iodine during late pregnancy, and the second group of ewes did not receive iodine supplementation during late pregnancy. When those ewes lambed, then one lamb received colostrum from its own mother, and one ewe received colostrum from, one lamb received colostrum from a ewe on the opposing treatment. So essentially, we could differentiate whether the problem arose within the lamb itself or whether the problem existed within the colostrum. And to cut a long and rather complicated application of this particular experimental design, quite short, regardless of the source of colostrum fed to the lamb, if the lamb is born to a ewe which received iodine or high levels of iodine supplementation in late pregnancy, then that lamb has a reduced ability to absorb colostral antibodies. So again, this gave us a focus in terms of where to go to next, and to look in detail at what is happening within the lamb. And another one of my PhD students, Fiona McGovern, really delved into this in great detail as part of her PhD thesis. And essentially, we were able to discover that when animals received iodine supplementation in late pregnancy, they produced offspring with an altered gut morphology, an altered thyroid hormone status, and alterations to the expression of various genes at the gastrointestinal tract level, which are important for antibody absorption. So we could follow right through the system from how we fed the ewe to how the lamb responded in terms of its phenotype, and then investigate what was happening at an intestinal, at a morphological, and at a cellular level within those lambs, explaining the phenomenon that we'd witnessed. So that was really interesting, was really detailed and component research. And I probably kind of figured out at that stage, I didn't want to be getting, I suppose, not dragged down, but getting absorbed with the minutiae of the detail. And I wanted to expand out my research to kind of look at a slightly bigger picture. And it was around that time that I started to get involved in, measuring and mitigation of enteric methane emissions from ruminant production systems. And, going back to 2004 and 2005 and 2006, when this funding was very, very freely available to research in this area. And unfortunately, once the economic downturn came, that basically resulted in a guillotine being brought down on the funding in this area. And it's only ramped up in the last 3 or 4 years. And luckily, in the intervening years, we've moved from this type of kind of rather labor-intensive rudimentary measurement equipment to some of these more sophisticated and automated pieces of measurement equipment. So the technology has certainly moved ahead. And we have had some really interesting results, and I've produced some nice findings, I think, in this area over the time. Starting at a very simple level, when we look at the quality of the feed we offer to our grazing ruminants, simply by increasing the quality of that feed, we can reduce methane emissions per day from beef cattle by 7% and methane emissions per kilogram of average daily gain by 22%. So in our beef production systems, essentially we're trying to reach a target weight at which those animals will be slaughtered. So the functional unit in that system then is essentially average daily gain. So we can reduce our emissions per kilogram of average daily gain by 22%. Simply by altering the quality of grass we offer to our animals. When we looked at the same type of work with dairy cattle, and this was some work conducted by Cathal Wims, who is now a research scientist in New Zealand. But in Cathal's work, we recorded a 13% reduction per day in enteric methane emissions from grazing dairy cows. And again, when we express that on a functional unit basis, we reduced emissions per kilogram of milk solids by 18%. And these are interventions that many farmers are making at the moment, and that have an important impact on farm profitability. The problem, however, from an accounting perspective, is it's almost impossible at the moment to capture these interventions at farm level in the inventory. So even though farmers are making these gains from an efficiency and from a reduction in environmental emissions perspective, we can't actually capture those gains in the national inventory at the moment. Our animals spend considerable periods of time indoors in this country as well for all sorts of reasons. When they're indoors, if we moved our diet away from a forage-based diet to a concentrate-based diet, including cereal grains and soybean meal and various other components, there is potential to reduce their methane emissions by as much as 42% per day, and their methane emissions per kilogram of carcass gain, again, that's the functional unit in this particular scenario, by as much as 60% per day. So again, very large potential for reductions here during the finishing phase. However, this can also present challenges, I suppose, from a marketing perspective and from a consumer perception perspective, because in Ireland, our products are very much seen as pasture-based, grass-based, and forage-based. And if we move towards very high-level concentrate finishing, it can alter both the reality and the perception of how our products are produced. And then the final point I'll make on the methane mitigation work we've conducted over the years is the use of various feed supplements. And again, I suppose if any of you are anyway in touch with the research or with the popular press in this area in the last few months and years, you'll be hearing a lot about seaweed and a lot about nitrate proxies and analogs. We conducted a lot of work in terms of oil supplementation. Perhaps the greatest success we've seen is with linseed oil, and some work we published just last year would show that by feeding pasture-based dairy cows linseed oil twice a day when they enter the milking parlour, we can reduce their daily methane emissions by 17% and their emissions per kilogram of milk solids by 20%. So there are a wide range of readily available technologies here to mitigate enteric methane emissions, but we need to come up with a mechanism where these can be accounted for and farmers can be rewarded for taking on these technologies. So the most recent, I suppose, reincarnation of our research activity has been around the whole area of production systems, and particularly the concept of multi-species swards and how multi-species swards can, I suppose, have an impact on a lot of the inputs and the externalities involved in livestock production systems at a national level and potentially at an international level as well. I started work in this area thanks to my colleague, Dr. Helen Sheridan, and Helen had been working on this for a number of years. She allowed me to become involved in one of her research projects, Smartgrass, a few years back. So essentially we looked at 4 different sward types and determined the impact of these sward types on animal performance. I suppose the big differences between the sward types, without boring you with the details, is that we reduced the quantity of fertilizer, nitrogen being applied to the swards as we increased the botanical complexity of the swards which were growing. And then when I got involved and started to measure animal performance, and we compared different sward types. The red column here is the perennial ryegrass monoculture. The blue is a 2-species mixture. The green is a 6-species mixture, and the yellow is a 9-species mixture. And the 6-species mixture was the one which provided us with the greatest success in this particular study. So compared to a perennial ryegrass sward, lambs suckling ewes which grazed the 6-species sward had a 2.4 kilogram higher live weight at weaning. So each ewe was suckling 2 lambs. So that meant each ewe was weaning an additional 5 kilos of lamb live weight at 100 days of age. So that's a really, I suppose, substantial improvement in lamb performance, which is good from a farm economic perspective. Probably what was most telling in that research was that the grazing the animals on the multi-species swards allowed us to reduce the amount of medicine we were giving to those animals to control intestinal parasites by as much as 50%. So when livestock graze pasture or graze grass, they're exposed to parasites which live in the digestive tract. And in fact, how we manage our grazing livestock can often increase that parasite challenge to the animals by grazing relatively large numbers of animals on relatively small areas of pasture at an individual time point. For many decades, we've controlled this challenge by using anthelmintics, a particular drug class which will control or kill the parasites. But in recent years, it's been recorded that the efficacy of those anthelmintics are declining quite dramatically. So I think this is a really exciting finding that compared to lambs grazing perennial ryegrass only, lambs grazing multi-species swards required 50% fewer drench administrations over their lifetime. And since that work with sheep presented such promising results, we've moved on into the beef sector, where a much larger proportion of our farmers are engaged in beef production. A much larger number of farmers are, producing beef, and it makes a much larger contribution to our national agri-food outputs. In this study, we had 3 sward types. We again had our ryegrass in red, our ryegrass clover in yellow, and our multi-species or 6-species mixture represented in green. And this work is only in its infancy and it's underway on the long-term grazing platform in lines. But when we look at the effect of sward type on steer live weight gain, and we compare the 3 groups of animals who have been grazing on these swards for about 18 months at this stage, we can see that the animals grazing the multi-species sward here in green, currently they're about 80 kilograms heavier than the animals grazing the perennial ryegrass swards. And that's an enormous difference in animal performance, mediated simply through the type of herbage or the type of grass essentially they're grazing on a daily basis. Helen has probably presented this slide in her presentation a few weeks back, but those swards were producing more feed for our animals. Our multi-species swards were producing more feed for our animals, despite the fact that they were receiving 60% less fertilizer nitrogen. And we're probably starting to think we can reduce those swards down to close to 0 fertilizer nitrogen without much, or indeed, if any, loss in terms of sward performance. But that's something we need to investigate yet. So at this stage, I think I'd just like to, I suppose, paint a picture of who we work with and how we're funded. So really, my work is part of a multidisciplinary team working across a whole range of areas, and I've only very, very briefly touched on a couple of those areas this morning. Our main funding sources and our main funders are listed on the left here, including the Department of Agriculture, Irish Research Council, Enterprise Ireland, and Enterprise Ireland is becoming an increasingly important funder of the research I do, as more and more industry partners are becoming involved in co-funding of our research. Teagasc are a funder through the Walsh Fellowship Scheme and also a very important collaborator, as are the Marie Curie ITNs. I think in recognising the challenge that exists, I've become more involved with global consortia and global initiatives. So my research team are aligned with the Global Research Alliance on Agricultural Greenhouse Gases, and we're members of the Global Farm Platform, which is an international grouping focused on sustainable pasture-based ruminant production. We collaborate right across national bodies, and I've just listed some who are currently collaborating with us, including the Technological University of Dublin, AFBI, and Massey University in New Zealand. And more, we're working, and I'm working a lot more closely with industry. And again, some of these industry partners who are contributing cash contributions to our research work, from large multinationals like Allflex, to new startups like the Cotter Crate. So I think that's a really important part of how we get our research out to the end user as well, is working closely with industry in the conduct and in the design of our experimental work. I'd just like to acknowledge all the colleagues, or many of my colleagues, who contributed to some of the work I presented this morning. I've listed many here. I'm sure I've left some of them out, so I'm trying to capture that by saying and many others. And I suppose the photographs represent 2 very important groups of people in the life of any PI or any research scientist. First, we have the postgrads and the undergrads who contribute to the research, and second are my own family members. I suppose to differentiate between the two, the group on the left are generally very, very happy when I leave work, and the group on the right tend to be happy when I arrive home from work. So it's important not to overlook either group for the important contribution they make. So Geraldine, I'll leave it there with that. If there's any questions, I'll try to deal with them, and hopefully I'm not too much over time.